we'll wait for 5 minutes, let people join, then we'll start. Okay, we'll start with our lecture. Deshmukh sir, are you there?
No, ma'am, Deshmukh sir is not there. I am there.
Okay, so shall I start?
Yes, ma'am, you can start. Yes, yes.
Okay, sure. So, good afternoon, everyone, and welcome to this insightful virtual lecture on chemokine axis and cancer.
Good afternoon, ma'am.
Yes. Please mute yourself. So we have arranged a virtual lecture on chemokine axis and cancer. So we are honored to have with us Dr. Amresh Mishra, a distinguished researcher in cancer biology. And Dr. Amresh Mishra, sir, is an ICMR senior researcher fellow at the School of Biotechnology, Gautam Budh University, Greater Noida. His research primarily focus on breast polyphenols. He has an extensive background in cancer therapeutics, computational drug discovery. To learn from his expertise today, so without further delay, I would like to invite Dr. Amresh Mishra sir to take over the session.
Thank you, ma'am. I'm audible to all of you.
Yes, sir. Yes, you are audible.
Okay. So thank you for the introduction and opportunity to give me a brief talk on my PhD topic. So let me share my screen. Okay, my screen is visible to all of you.
Yes, sir, it is visible.
Okay. So today's talk is on chemokinesis and cancer. Uh, please, this was my PhD thesis topic. So I will brief you that what is the chemokines and what uh, chemokine axis and what is the role of these chemokines and these axes in cancer. And my PhD topic was chemokinetics, role of chemokinetics, particularly in breast cancer. So why I chooses some particular chemokine ligand and receptors which make an axis, and why I choose particularly breast cancer. What is the rationality behind it? What was the novelty behind it? What what was the challenges? What is the limitation? So all I will talk in this uh, lecture. So all we know that chemokines are small proteins which uh, like if you have for example, I'm giving just an example. Uh, if uh, you have fever, then in case of fever, there will be the inflammation and many immune cells get activated. So there are many chemokines who secreted uh, during various immunological activities, inflammations. But uh, if we talk about in terms of disease in cancer, so there are several chemokines who are sorry, who are highly upregulated in case of cancer. And during my PhD, I choose uh, three chemokines. One chemokine was a ligand, and two chemokines proteins were act as a receptor, and all three together makes an axis, and this axis is highly upregulated in case of breast cancer particularly. That's why I selected this particular chemokines and the ligand and receptors which makes an axis, and selected breast cancer because when I go through the literature, uh, I have been found that this axis has the potential to target uh, for breast cancer. So, my PhD objective were divided by two parts. First was the in silico bioinformatics part, and the insight I got from the in silico, the bioinformatics part, validated further validated by the in vitro experiments. Uh, so I also check it using the different breast cancer cell lines like MCF7 and perform all the experiment. One thing uh, was very rational and novel during my PhD study that uh, I I target breast cancer cells as well as the breast cancer stem cells. What is the breast cancer stem cells? Breast cancer stem cells is like highly resistant even for chemotherapy. Uh, if suppose if one person has any cancer, so we are supposing that uh, X person having breast cancer, and we are giving chemotherapy to her. So if the in the breast cancer or in the tumor, there will be population. If there will be population presence for breast cancer stem cells, if breast cancer stem cells population will be present, so it most probably it will register the chemotherapy or any kind of therapy. So targeting this particular pathway and my compound, which I have been selected using computational analysis. So my compound was targeting this pathway, and through this pathway, the compound was targeting not only the breast cancer cells. Uh, that compound was very effective and very promising to target breast cancer stem cells also, which is the real culprit for progression and metastasis of breast cancer, relapse of breast cancer. So that was the novelty. So when you select any study, when you select any protein, any target, uh, it must be any rationality behind it, any novelty behind it. So chemokine has already defined that these are the small proteins and uh, guiding movement of immune cells. Chemokine divided in four classes. CHC chemo, what I selected was CHC chemokines. So uh, basis of the residue uh, chemokines having, so we divided into four parts. So my chemokines having CXC residues, cysteine. So it was belongs to CXC chemokines family only. There were other three families: CC chemokines, C chemokines, and CXC3 chemokines. As I told you before, that I selected three chemokines. One was the ligand, and two were the receptors. Every ligand effective only when uh, the ligand will bind to the corresponding receptor. Every ligand has their corresponding receptor. My ligand has this. Please turn off your all of. I'm requesting you that please turn off your mic and video also. If you have any question, you can write in the chat box. Uh, I'll try to answer all of your query after the this session. So, all the chemokine receptors known as G protein coupled receptors, I think if you are not very familiar, at least you listen this GPCR receptor or GPCR name or G protein coupled receptor name somewhere. If you are pursuing your masters, or I'm assuming that all diversity in the participant, that some of you are doing your bachelor, some of doing your masters, some of your uh, research work or project fellow. So I think most of them aware or at least listen about G protein couple receptors. And I'm assuming that all of you know that what is the chemokines. You are not having that much of uh, like that much of familiarity that what is the chemokine receptors class. So, yeah, but you have the basic idea. I'm assuming from you. So there are many signaling pathway, uh, including MAP kinase and NF-kappa B pathway. So chemokines play a key role, uh, and that's why targeting chemokines and chemokinexis is very rational to any disease, including cancer or particularly if we talk about breast cancer. So, as I told you, that chemokines has, uh, in terms of cancer, chemokines have a role in metastasis, role in progression of cancer, even in tumor microenvironment. So, how chemokines have the role in tumor cell progression or proliferation of tumor cells. So, by activating these signaling pathways, MAP kinase, NF-kappa B signaling pathway, by activating these pathways, uh, chemokine trigger the cell proliferation, tumor cell proliferation. And this proliferation leads to tumorigenesis, tumor microenvironment, and tumor progression. So, yeah, regulating these pathways, they are very uh, crucial target. This makes very crucial and novel target to uh, select this pathway particularly. Yeah, they have the role in tumor angiogenesis, tumor cell proliferation, tumor metastasis by activating these pathways. So now we are coming that how does chemokinesis plays a role in cancer progression. So I selected CXCL12, which was a chemokine ligand, and CXCR4 and CXCR7, and these two were a receptor. So when CXCL12 binds with these corresponding receptors, CXCR4 and CXCR7, it makes an axis and it is highly upregulated in case of breast cancer particularly. So this axis uh, makes activated all the down-regulated and up-regulated signaling pathways and has played a key role in terms of, if we say, tumor progression, metastasis. So that's how this axis may and then further uh, play a role in cancer progression. So these are the different axes like CXCR4 and CXCL12 axis, CCR7, CC, CCR, CXCR. So I defined that uh, on the basis of the classes, on the basis of the residues, we divided the chemokines. So that's why you see here that there are different chemokine and different chemokine axes. So this class is CXC. So CXCR4, CXL12 axis, CCR7, CCL19, and CCL21 axis. Here you can see there are two ligand and one receptor. So these two ligands binds with this receptor and get have the key role in tumor progression and metastasis. This another class of chemokines which make axis CCR5 and CCL5 axis. So this axis has very rational to target in any cancer, including breast cancer. So there are many inhibitors which already have in the clinical trials also. So many in silico, in vitro, in vivo result are there. You can find in many literature. But apart from this, in silico, in vitro, and in vivo, they has clinical relevance also. They already in the clinical stage. Uh, So CXCR4 is one of them, which already ongoing in the clinical trial to inhibit CXCL12. We can block the interaction of CXCL12 with CXCR4, and this interaction block we can uh, like stop the progression, or at least uh, try to give the therapeutic importance the cancer patients, including breast cancer patients. We can stop the uh, cell migration or metastasis, we can say. CCR7 inhibitors is already proposed for clinical trials. CCR5 inhibitors. These all the inhibitors. Uh, this is primary CCR4 inhibitor primary used to treat HIV infection. Later on, we find that okay, these chemokines also have relevance in various breast cancer. So, and like proposing chemokine inhibitors alone may be not very effective, but when we uh, when we give these inhibitors uh, together with the chemokine therapy to cancer patient, it it, it has been already like proposed, and practically I mean you can find lots of literature. uh, Outcome was very effective when we give chemotherapy along with these chemokine receptors. So this is very promising, very effective therapy to give in combination with the uh, chemotherapy to different cancer patients. So together with the chemotherapy, we also giving the chemokine inhibitors to uh, expecting that the patient will recover, or uh, the doses of chemotherapy may be reduced because of this uh, chemokine inhibitors, because the chemotherapy has uh, lots of side effects. So if we reduce, then it will be very good for patient. So this is already in the literature. Yeah, I mean, apart from its promising and effectiveness uh, to target various cancer, there are challenges also. There are limitations also. Uh, After my PhD, I I was thinking ki uh, this axis or these chemokines are highly upregulated in case of breast cancer. So, what if we propose an uh, instrument so that we can we have to validate that this person having the breast cancer, and this person uh, uh, don't have the breast cancer, on the basis of upregulation or high upregulation of these markers. So can we use these chemokines as a marker? So I propose to ICMR that I want to work to develop something, develop a sensor, so that we are able to know, we are able to diagnose uh, that this person has the breast cancer, this person don't have the breast cancer. But there were limitation. My proposal got rejected. Why? Because chemokines are small proteins and highly upregulated not only in case of cancer or any other disease. Uh, I mean, I was proposing for breast cancer only, but breast cancer के केस में ही थोड़ी ना upregulation होता है. There are many several uh, even uh, पहले मैंने एग्जांपल दिया फीवर का, है ना? So if you have fever, there are lots of chemokines who are highly upregulated. So suppose if a person having the breast cancer, if person having a fever, and uh, I I just use these chemokines as a marker molecule. So both will be detected that both person has the breast cancer. So proposing an idea, uh, we should very well aware that what is the limitation, what is the challenges. So yeah, we all uh, don't have very emotional about that. No, this is my PhD topic and I have expertise. Uh, I'm working very hard since last 4 years. I'm working on it. So uh, apart from this side, we also have to criticize our work. We also very well aware about the challenges, the limitation of it. So yeah, that was the example of mine. Yeah, no doubt that future of chemokines and chemokine antagonist is very promising, but there are limitations, there are side effects also. So we have to tackle it down. We have to reduce it either from the doses or either from working on another chemokines inhibitors. Yeah, as I already told you that there are we already given the combination of uh, chemotherapy. So together, uh, the chemokine inhibitor and uh, chemotherapy, yeah, biomarker development. I already told you the example that we can develop using this biomarker. What what was the lacking behind on my proposal? I should make this chemokines marker is okay, ठीक है, breast cancer के केस में highly upregulate करते हैं, but I don't have to rely completely on these markers only. I have to use some other biomarkers so that it could be validated, and those biomarkers only particularly for breast cancer. But if we use multiple biomarkers for one device or one test, so it will be increase cost, the time. So we have to choose the biomarkers. Please turn off your mic. We have to choose our biomarkers very precisely, which should be uh, uh, like have the relevance, have the relevancy behind it. Yeah, there are many clinical trials. Like I already told you that how many chemokines inhibitors are ongoing in clinical trial and uh, case studies. Okay, I was muted. Uh, I'm audible to all of you.
Yes, sir.
Okay, so there are many examples that how many chemokines inhibitors, different chemokine inhibitors used in various uh, cancer. like CCR5 inhibitors using in breast cancer. I'm not able to move my slides. Okay,
Dr. Amrish, can
stop.
you hear me?
Yeah, I can hear you.
Are you continuing now this one?
Uh, Sir, but let me complete. There are
Okay,
two,
okay,
three slides left.
okay,
Then,
okay.
yeah.
Sir, are you able to move the slides?
Uh, Wait, I'll let you know. Okay, yeah. Now I'm able.
Okay.
So CCR7 inhibitors in lymphoma. So yeah, uh, this is the summary of key point of my lecture. And if you have any questions, you can ask me or you can write in the chat box so I can answer. Yes, sir, please ask your questions.
Yes, Prakash sir.
Dr. Amrish, can you hear me now?
Yes, sir.
Hello?
Sir, yes, sir.
Oh, wonderful.
Yes, sir, I can hear you.
Now, are you continuing this work now are you continuing this work in Gautam Buddha University or after PhD you have stopped?
As I am
Yeah.
currently working here, as sir, I'm audible to you.
Yes, I can
Yeah.
hear you.
So currently, as I am working as an assistant professor in Parul University, so
Hmm.
there is much other responsibilities like
Teaching.
teaching workload
Okay,
and other administrative responsibilities. So I'm not able to continue, but I proposes some good ideas to the
okay.
funding agency. Even yesterday, I submitted one um, uh, project to ICMR. So when I grant the project, I'll have less teaching opportunity and no other administrative opportunity. Then I can explore and continue my PhD work. So yeah, I want to continue and I will continue, but later on. Currently, I'm not doing research work.
Wonderful. This with relation to colorectal cancer, you will have the different kind of axis, right?
Yeah, even in colorectal cancer, when I was going through the literature to prepare my research proposal and defend my synopsis in front of my PhD committee. So yeah,
Okay.
I found that there has been uh, a role in colorectal cancer also. Many chemokines has the roles.
So the same, no, because colorectal cancer can be treated using probiotics, and this is what actually we are looking for. So
Yeah.
how, with your knowledge or with your experience of during PhD in colorectal cancer, this inhibitors, if we use as a control, will be useful. In other words, what is your opinion?
Yes, sir. I mean, definitely, sir. But we have to select those chemokines
Ah,
like I selected CXCL12, CXCR4, CXCR7 because
okay.
when I go through the different literature, I found that these three chemokines are key chemokines which are highly upregulated in case of breast cancer only. They has the
Hmm.
role in other cancers also, but in case of breast cancer particularly,
Hmm.
they are highly upregulated. And my motive is to choose this chemokine axis is not only to target breast cancer, but target breast cancer stem cells also, which I
Okay.
already uh, like when I perform my in vitro experiments, I already have been found that okay, this using this chemokinetics uh, and my compound was a dietary polyphenols. So that compound is targeting breast cancer and breast cancer stem cells through this chemokinetics. So in colorectal कैंसर के केस में हमें देखना होगा कि कौन कौन सा कीमोकाइन एक्सिस है जो रोल प्ले कर रहे हैं एंड to I mean apart from this we also have to very well like very well aware that to how to screen and what compound we have to screen out so that we can target colorectal cancer through this chemokines or this chemokine axis.
What type of cell line you use for your study?
MCF7. MDA cell line also used, but uh, that cell line is like very tough to maintain. So we go with the MCF cell lines. So we proposes two cell lines. One was optional, but yeah, we go with the MCF7 also.
Thank you. Thank you.
Very nice, Amresh. You have
Thank you,
given
sir.
a very excellent talk. There are few more questions in the chat box.
Let me
Akanksha will read that questions and you can
Yes, sir.
answer that question. Again, I will tell you, we have recorded your lecture. It will be uploaded on the YouTube of Yum Bio Society. It will be circulated throughout the India. And it is a very good practice that the research of newly coming PhD researchers should be known to all Indians, not only in India, outside the India also, what work is going on.
Yes, sir. Very
in
useful
cancer.
work.
Correct, breast cancer. It will
Yes,
be
sir.
useful to many others. Really, I'm happy you have
And
participated.
colorectal
Pardon?
cancer, which is actually a microbiology through probiotics.
Yes. Yes, yes, yes. Yes. So, I appreciate you. Pardon, 1 minute. I appreciate you as well as I appreciate Akanksha also for organizing such a beautiful lecture series.
Sir, unable to hear you.
Hello?
Yes, sir.
Hello?
You are audible.
Ah. So I appreciate Amresh
Thank you, sir. Thank you, sir.
as well as I appreciate Akanksha. You can hear me?
Yes, sir. Thank you so much, sir.
Yes, for taking a responsibility of such a beautiful lecture series. Now, Akanksha will read the questions which are given in the chat box.
Yes,
Okay?
sir. So one question is from Adil. So first question, how we can counter specific cell so that other cell cannot be harmed? Amrish sir, are you audible?
What's the problem, Amrish? Amrish, you are unmuted, but we
I guess there
cannot
is some
listen.
network issue with him.
Okay. Okay, let us wait for 1 or 2 minutes. In the meantime,
Yes, sir. Yeah,
I
he'll
also
rejoin.
appreciate Kanan also. She has asked many questions,
Yes.
correct? Yes. So, Kanak, not Kanan, Kanak. Kanak has asked question, Prakash Alami. You can proceed.
Hello. Sorry, my internet connection was. Can you repeat? Can you
Yes, sir. How we can counter
repeat the
specific
question?
cell? Yes, how we can counter specific cells so that other cell cannot be harmed? Sir, if
So
I'm not audible, you can read the questions in chat box also.
yeah, I'm opening the chat box.
Yeah, sir, sure. From that you can answer them.
Okay, so it was from Adil. Adil have two questions, that how we can counter a specific cell so that other cells can't be harmed, and can we modify a gene in such a way so that they can be helpful in making protein? Can we modify a gene in such way so that they can be helpful making protein which will help over the cancer? So answering your first question, that yeah, we can counter specific cell so that other cell can't be harmed. Like in using nano technology, we can counter these specific cells. Suppose there are tumor cells population, so we can bind the uh, uh, nanoparticles to particularly for tumor cells, and then we can identify that okay, this much of tumor population or cancer cell population is there in the body or there in the particular organ, and then we can identify them and target them particularly so that to make sure that no other cell is harmed. And answering your second question, that can we modify a gene in such a way so that they can be helpful in making protein? Yes, we can do, but there are again in nanobiotechnology or in genetic engineering, there are many limitations of also. They are very precise, they are very effective, they are very promising, but at the same time, they have limitations also. They have very like uh, ethical limitations also, as well as uh, the like I told you that there are many chemokine inhibitors which have very promising, but again they have uh, the side effects also, and they may be effective for short time of period, but later on they show the toxicity or chemotoxic to for to harm other cells. So yeah, we can modify using CRISPR-Cas9 technique and many other recent genetic engineering. We can modify a gene. So there is other question from Prakash sir, that what type of inhibitor you are looking for, which will be very specific without any side effect. Sir, uh,
Yeah,
those
most of these
inhibitors,
my questions are already discussed. So you can
yeah,
go
yeah,
to the people online, those who are there, if they have any questions.
yeah.
Yes.
Kanak,
Oh.
Kanak is asking that as we know, cancer treatment is not that affordable to all the people. So what are your contribution? Okay, my contribution is very small because first of all, I I performed in silico and in vitro study, but using my study insights, it may be proposed that okay, we should go further for in vivo analysis, and then okay, this chemokine exists particularly very effective to target not only breast cancer but breast cancer is, stem cells also. So we can go further with the clinical trials and the case studies. So yes, my contribution will be there if using these my study insight will go further in vivo clinical trial and case study analysis. So it is a strong possibilities that uh, there will be another chemokine inhibitors, like previously I told you that some few of chemokine inhibitors proposed and already in the clinical trials. So these chemokines which I have been selected for, particularly breast cancer, so these chemokines also proposed as an inhibitor, or this axis inhibitors, this chemokine axis inhibitor could also be been proposed. Then my contribution will be practical uh, and translational applications. As of now, uh, very hypothetically, contribution is there. Ki, okay, this insight I have from my study, and this study should be reflect for further study, and further study must be needed. So yeah, my contribution is there, but very small.
Okay, okay, Akanksha, now you can continue.
Yes. So, thank you, sir, for this enlightenment session on the role of chemokines axis in cancer. So, we have a lot of information today. So, we have understood a potential cancer treatment strategies today. So, and I would like to extend my gratitude to all the participants for joining us today. Your engagement and questions made this session more interactive and meaningful. And special thanks to Dr. Arvind Deshmukh sir, President Microbiologist Society India, for making this virtual lecture possible. And once again, thank you, Dr. Amresh sir, for sharing your knowledge with us. We look forward to uh, have more informative sessions in the future. Thank you so much, everyone, for joining. So have a
Thank you
wonderful day
so much.
ahead.